Now let's study about molecular imaging. Molecular imaging means we use molecular imaging probes. We have seen lots of fluorescence molecular probes, but fluorescence has the same limitation of light penetration as in optical imaging. So let's think about radioactive probes. Radioactivity is unstable nucleus either decompose or fuse, and they change its structure and generate small particles or strong electromagnetic energy beams. The common particles of this radioactivity is alpha particle or neutrons or protons and beta rays. Beta is the electron beam, and high energy of short wavelengths of a gamma ray. The radiation pattern is different according to different atoms. Here are some atoms which generate gamma rays. Gamma ray has a shorter wavelength than X-ray, and the strong energy can generate images. Among these known metals, seems like a 99M technetium has a reasonably short half-life, about 6 hours, and this has been used a very popular probe for gamma ray imaging. Technetium itself is a metal ion, so usually it's not easy to use as is. And usually we make the complex with a ligand, and in this case, this ligand is called as HMPAO. The complexation of these two units make stable probes for clinical imaging. So, for example, if this kind of a molecule enters to the body, and it generates stronger energy than X-ray, it can show the position of those probes. In X-ray images, we utilize the external X-ray light, and some part of the light is absorbed by the bone, and only penetrated light we detect and then construct the images. But in gamma ray images, we just detect those gamma rays generated by probe already localized inside the body. This image shows the probe locations inside the lung. Advantage of this gamma ray is we can image quite deep because the gamma ray has very strong energy, but in general, the resolution is quite low. Now let's look at SPECT. SPECT is a single photon emission computed tomography. Here, single photon means gamma ray, CT means we generate three-dimensional images. So altogether, gamma ray based 3D image, this is SPECT. SPECT utilize this kind of multi-angled cameras and then collecting multi-dimensional images to construct tomography images. This is the SPECT image obtained from the lung. As the spatial resolution of SPECT is not so good, usually we use X-ray as a guide for the anatomical structure, utilize those combined images. Altogether we call SPECT CT. SPECT has CT inside, but second CT is X-ray CT. By using combined SPECT CT, now we can tell which part of those probes, and this is guided by these X-ray images outside. In this animal experiment, we can see the high resolution of X-ray CT image, and then we can monitor those localization of those probes along those gastrointestinal tracts. This kind of SPECT CT images can be used for localizing the specific site of cancers. This example shows us the position of thyroid cancer. But in case the radioactive molecule is metal, we need to use a ligand, and also we should add another modality of specific delivery of those probes to the target site. How about iodine? Iodine has variable isotopes, and the most common stable isotope is I-127. Thyroid is a gland which makes hormones which contains iodine. That's why if you inject iodide into the body, the iodide tends to go to thyroid. If you inject iodide 131, which generate gamma ray, it can be used to image thyroid cancers. Again, combined together with the X-ray CT, now we can see those locations and then localization of iodide the positioning. More important thing is, compared to metals, iodine is easier to incorporate into organic molecules. What is this molecule? This is a tyrosine. And here, it's possible to introduce iodide utilizing organic chemistry. That means variable organic molecules can be generated for spec probes utilizing iodine-131 or another gamma-ray generating isotope, iodine-125. Then you can imagine any kind of cancer targeting organic molecules. You find the specific positions to introduce iodine. Then you can make imaging probes to send the molecule to the target positions. Now let's think about a PET. PET is positron emission tomography. What is a positron? Usually we hear the radioactivity as alpha ray and beta ray and gamma ray. That means that these three are most common radioactivities. Sometimes very strange particles also can be generated. One of them is a positron. Positron is very similar to electron, but electron has a negative charge, but positron has positive charge. It's kind of an antiparticle. Positron and electron can be generated by collision of a very strong gamma ray, and collision site is here, and electron and positron inside the magnetic field, because of their opposite charges, they make different directional movement. In reversible process, if electron meets positron, they generate two gamma ray photons. Some special radioactive nucleus, they can generate a positron. Positron diffuse out to the environment, and when they meet with the electron, now they annihilate, means disappear, and generate two photons of gamma ray, and these two photons, they fly 180-degree opposite directions. Among these nucleus are fluorine-18, carbon-11, and oxygen-15, and nitrogen-13. The half-life of oxygen is about 2 minutes, maybe a little too short to be used for imaging probes. Carbon-11 is about 20 minutes, and F-18 is about 2 hours. Then you can imagine F-18 is the most convenient probe for PET imaging. We can use also carbon-11, but this half-life can make the chemist and also the imaging scientist quite busy to make the probes and also acquiring the images. Once the probe is localized in the body, it generates a positron, and by meeting with the electron, it can generate two photons. The gamma ray fly into the 180-degree direction and they hit the detector at the same time. In SPEC case, it was a single photon, but PET case, it generated two photons with the defined angles of 180 degree. That's why analyzing the original position of those probes is much easier than SPEC. This makes PET in general higher resolution technique than SPEC. Let's think about the PET probe, fluorine-18. Do you have any idea where the fluorine-18 can bind? Iodide case we have seen, it has an intrinsic affinity to thyroid, and we could use iodide for thyroid imaging or thyroid cancers. Fluoride? We utilize fluoride for toothpaste. Why? Fluoride can bind to tooth bone, and we believe it can make the tooth stronger. Yes, fluoride has affinity to the bones. If we inject fluoride-18, it can show us the bone structure in the body. This image can be similar to the X-ray, but the mechanism is different. Maybe these two techniques we can say complementary techniques. Most representative and popular PET probe is glucose derivatives FDG. FDG is a fluorodeoxyglucose. Glucose is the most common biological energy source. High glucose concentration means high energy consumption is happening. Let's think about the brain. Brain's weight is only 2% of the whole body, but energy consumption is about 20%. Actively energy consuming organ brain has very high glucose concentrations. Even inside the brain, those the dynamics of the glucose concentration change is very fast. For example, if we give some visual signal and then we can monitor which part of the brain is activated. Utilizing this PET technique with FDG as a probe, we could map which part of the brain is really visual area. If you compare two brains, one is a normal brain and one is cocaine-addicted brain, then brain function is reduced. That's why glucose concentration is much higher in active brain and not in this suppressed brain. Not only for the normal functions, cancer cells, they consume a lot of energies, especially by high rate of glycolysis. That's why FDG has been used to detect cancers. In this image, we see high concentration of the glucose in the brain, but at the same time, there are a lot of spots in the body, and these spots represent cancer part. Combined together X-ray CT, and we can localize which part of the body has these cancer cells. One of the limitations of FDG is involved in too many different phenotypes. FDG can be localized in highly functional area, or in cancer, or in inflammation. So in many cases, FDG image itself is not good enough to make specific determination. Then it would be ideal, in each case of phenomena, we have specific molecular probes. Let's think about one more example with this amyloid plaques. Amyloid is a denatured protein, and if they aggregate, they interfere with the normal function of the body. Alzheimer's disease is one of the cases, and if you have an amyloid plaque in the brain, and functionally if you have dementia, we diagnose the patient's Alzheimer's disease. Some good numbers of these beta-amyloid specific imaging probes have been developed. In the beginning, these probes are all fluorescent versions. To utilize these probes into PET probe, what do you need to do? Either you introduce carbon-11 or F18. One of the early compounds is PIB. PIB is a Pittsburgh compound B. And starting from the thioplavin, which has a positive charges here, and by removing these charges, and then introducing carbon-11 here, they made first PET probe. When this compound is applied to Alzheimer's disease patient and control person, now you can see much stronger signal from Alzheimer's disease patient. And this shows the possibility that we can detect Alzheimer's using PET without any surgery. Next version compound is the F18 version of FDDMP developed by UCLA scientists. In their test, normal people has a low signal here. When the cognition function is impaired, now the signal becomes getting stronger and stronger. And in this study, the PIB and FDDMP has been compared together, and this side is the control and this is the AD. Selectivity-wise, seems like PIB is better than FDDMP. But you remember, the lifetime of carbon-11 is only 20 minutes, and F18 is about 2 hours. So F18 may be much more convenient to prepare the reagent and acquiring PET images from patient. So it would be quite desirable to have more selective probes using F18. Scientists from UPenn, they developed AV45 with F18. This probe showed quite good contrast between AD versus control patient. And this compound is the first FDA-approved PET probe for beta-amyloid for Alzheimer's disease diagnosis. As you can see here, if you have any organic molecules for specific probes, by incorporating carbon-11 or F18, you can change your molecule into PET probes. Then you can visualize the specific location of your target even in live human body. That is the power of SPECT or PET technologies.